concentration, atmospheric concentration of CO2 of gases is dominated by nitrogen, which is about 70%, about 16% oxygen. 400 parts per million represents 0.04% of the total complement of gases in the atmosphere. So we're dealing with very small amounts, but of course, as we know through the heating effect that this greenhouse gas, one of the greenhouse gases being CO2, has, is it warms the atmosphere, and that generates different weather patterns, flooding, droughts, storms, weather at inopportune times, and so on. What we're focusing on here, though, is the actual effect of the increase in CO2 itself, the primary cause, and thinking about that in terms of plants. So I'm a plant biologist, and one of my favorite things is these things, stomata. Stomata are the solution to the plants colonizing the land many billions of years ago, and finding out that the land was quite dry, and if you didn't have a cuticle, you would dry out. So the solution is to have these little pores, and these pores, stomatal pores surrounded by these pair of guard cells, allow CO2 to get in across the cuticle, the waterproof cuticle. The other problem that plants have, of course, is that if you open your stomata to let the CO2 out, the water can come out. And so you've got this tension between taking in carbon dioxide for photosynthesis, the food of the plant and the driver of most of the life on Earth, and then the losing of water and potentially being droughted. And this is a tension that plants face all the time. And we'll come back to that when we look at the FACE experiment. Of course, on a basic level, we're looking at the carbon cycle, and of course, photosynthesis and the biosphere carbon store is crucial to that. And BIFoR is looking at photosynthesis, also, as you'll see, respiration, decomposition, and the carbon stores in the soil and what happens to them. We're also, through that water loss, also looking at that intersection through those tiny little pores of the carbon and the water cycle interacting through the contribution of plants and trees, specifically to water loss and transpiration, and more water then accumulating in the atmosphere. So the carbon and water cycle intersect through these little pores, and putting more carbon dioxide into the atmosphere is going to affect that fine balance, and indeed the decisions that plants make about whether to take in more carbon dioxide, or and if they do, what to do with it. And that's the crucial questions that we're trying to answer. So if I just come out of that for a second, and I'd like to just take you to Google Earth, which is obviously publicly available, and you can do this yourself. So there we are in the UK.
That looks great.
Is that okay, Jay? Can you see that?
Yeah, all good.
So if we zoom in, I'm sitting here in Birmingham in my office, looking out at a willow tree, actually. And if we zoom in just north of Birmingham, and in fact just west of Stafford, what we can see, if we find this place called Norbury Junction, which is an old junction of two canals, the Shropshire Union Canal being the main one that still exists. You can see this patch of woodland here, and actually something intriguing, some white stuff down here in the bottom left of this woodland. If you were to zoom in a little bit further, what you would see in this woodland were some rather strange structures. You can see 6 rings of pylons in this woodland. And this is actually the BIFoR FACE experiment. What we're doing here in these rings is we are squirting, and I'll explain how that works when we get onto the virtual tour a little bit more. We're pumping in carbon dioxide into 3 of these rings to hit 150 parts per million above the ambient CO2 at the moment, anticipating what we think the CO2 levels, the atmospheric CO2 levels, will be in 50 years' time. So it's almost like a little time machine. So 3 of those rings, they have elevated CO2 going in at 150 parts per million, and 3 of those rings have simply air at the same temperature as the rings. Just to give you a sense of scale, you can see some little cars here down in the car park. Okay, and if you look at one of those rings, We'll get a closer look at them later when we go on the virtual tour. These pylons about 25 meters high, and the ring is about 30 meters across. You see the general shape of this is a rough ring with oak trees, mature oak trees inside, and a central pylon, which, as you'll see in a moment, gives us access to the treetops, which is crucial for the research. And basically, the simple thing is we're squirting carbon dioxide into these and not into some of the others. And we are asking the question, do the trees take in more carbon? And if they do, what do they do with it? So basically, we're measuring almost everything. As you can imagine, this is unrestricted. And the reason for that is if you do it in a greenhouse or you do it in a growth cabinet, there are many other restrictions to the plants, but these are natural plants growing in a natural environment. And so that's the best way to understand what our ecosystems, what our temperate oak forest ecosystem is going to look like and how it's going to respond to this elevated CO2 in 50 years' time. And that, of course, will inform models that help us react and protect and respond to those changes. Looking down here at the things I flagged up earlier, this is the houses that you'll see closer up shortly. These house the control systems and the computers where the technicians can oversee the CO2 going into the rings. And these are the CO2 tanks, which contain 120 tons of CO2, which is processed, as you'll see, and piped to the rings. You can just see one of the pipes going through the forest there, and then distributed into the rings as required, depending on wind speed and how the experiment's going and so on. So that's Google Earth, which shows you it as it is today, or the last time the satellite flew over there. So if we go back to the actual talk, and I put that in just in case the internet went down, but obviously it didn't. And here's another shot in an image of the pylons. This one here on the bottom, on the right-hand side, you can see the ring that the pipe of CO2 is taking to the ring, which goes then to feed the pylons. This is a close-up, and as I say, when we walk in virtually, we'll see more of this. Here's with the pylons on the outside of the ring, and here is the ring that carries the CO2 from the control desks to these, you can't quite see it, but these blue valves are the ones that are controlled by the computer. And what they do is they let the CO2, each pylon has two of these black pipes hanging down, which are simply pierced with holes. And depending which way the wind's blowing, the computer will allow CO2 into this pipe, which then just blows out across the wind into the forest to be maintained at the appropriate level. Of course, the pipes on the other side, if the wind was blowing into the picture, the pipes on the other side would be closed. And it's very reactive and tracks it very, very accurately. Now, the experiment's been running for a number of years now, and the first data has started to come in. And the first data, this was generated by a PhD student, Anna Gardner. You can see her at the top of one of the pylons there in the central tower, which we'll come to later. And she had to climb the trees every day for 2 years and measure the photosynthesis at the top of the pylons, at the top of the canopy of the trees. You'll get a closer view when we go on the virtual tour in a moment. So Anna worked really hard on this for 2 years, and you see this data from June, July, August, and September from one of the years that she did it. It was the same for both years. You can basically see that the blue rings, the blue lines is the photosynthesis in the control rings where the CO2 is ambient, and the red line is the CO2 where we've added 150 parts per million, or 0.015% more CO2 to those rings. Now, I think it's clear that you can see over the summer, the red line is above the blue line. I should say this axis, the y-axis here, is the rate of photosynthesis. So the bigger that number, the more CO2 is being taken in. And this is 7 o'clock in the morning, and this is 7 o'clock at night. So clearly you can see there is more photosynthesis in the middle of the day when there is more light for the leaves to use as energy to capture that CO2 and make it into sugars. So that's the breaking news, if you like. That paper's been published, and that's really important because that sets the scene for what we do next and how the experiment goes forward over the next, hopefully 10 years, where we're looking now at what happens to that carbon. We know the trees are taking it in, But what happens to that carbon? They can make, as I alluded to with the stomata, they can make various decisions about what to do with it. Now, this is a picture of pine trees that have been grown at different levels of CO2 in a greenhouse. And you can see, if we give those pine trees more CO2, they grow bigger. But of course, these pine trees have been grown in a greenhouse with everything they need, all the nutrients they need, all the water they need, they've been protected from pests and so on. So the CO2 there is the limiting factor. Now, if I said to you, I'm going to give you a better diet, I'm going to give you more food, and I gave you a bag of sugar and a spoon, you'd quite rightly say, that's not a balanced diet. I need vitamins, I need fiber, I need fat, I need carbohydrates that aren't just processed sugar. And that's essentially exactly what's happening to the tree. It's taking in 30% more carbon, it's making 30% more sugar, but it can be limited by other things, such as other nutrients and other decisions it has to make. And that's why it becomes quite a subtle set of experiments. So we know the CO2 is being taken up by the trees. What are they going to do? Are they going to regulate their stomata differently to save water? Are they going to reproduce more? Are they going to defend themselves better? What's going to happen to the local ecology as different plant species respond in different ways? Are they going to grow more? Are the roots going to grow more? Or importantly, is a lot of that carbon going to go into the soil and stimulate the rhizosphere, the world around the roots, something we know almost nothing about. So, okay, so I'm just looking at the chat and we don't seem to have any comments or questions at this stage. Feel free to make a comment or question if you like, and we'll pick it up where appropriate. But I'm now going to take you to the virtual BIFoR, so you can have a look at some of the things that I've been talking about and what we're doing about it. I've put the link to this site in the chat, so you can go there at your leisure. Basically, what I'm going to talk you through today is that number one of that, a virtual walk through the whole forest. And there are also a set of education resources used by local schools looking at fungal growth, tree growth, and transpiration under elevated CO2. But I don't propose to go through that in detail today, although you're welcome to look at it and comment on it as you see fit later on. Somebody's asking what the enriched CO2 is in August. Again, that was we'd have to, and we can go back and look at the paper, but this would probably be because there'd been some, as I said, other things other than just the light and so on might be limiting, and the CO2 might be limiting. So it could be there was some poor weather or some herbivore attack or a cloudy period or a low temperature period. Although having said that, you don't see that particularly clearly in the controls, but that's a good question, and we could look at that in more detail by looking at the ancillary data back in the papers. So this is what we're going to do virtually. Now, if later on you want to come back to this and you've got a VR headset or something, it'll be even more spectacular for you. But I'm going to take you virtually to the opening of BIFoR, the gate, and then we're going to walk through the site. We're going to see the gas tanks and see some videos about the gas tanks. I'm going to walk you through the forest, and we're going to go into one ring and see some of the experiments, and then look at some of the experiments in another ring. And then we'll finish at the top of the main tower where you saw Anna's picture right at the beginning. So without further ado, I will, if I can just escape from that. And now I will go to the virtual BIFoR itself. So if you click on the links, it's done in a thing called ThingLink. And you can see, if I just roll around, you might recognize that was the car park where I showed you the cars. I'll make it go, if you had your virtual headset, if you clicked on that and set it up, you would be able to see this in glorious 3D vision rather than just a flat screen. I'll make it full screen at the moment. That's the car park where you saw the little cars. That's the road up to Norbury Park. And basically, as you walk through, arrows take you to the next stage, and the numbers give you some information. And I won't propose to go through every one of those. You can have a look at some of those yourself. But this is the main car park. Those are the offices where the technicians control things, and all the computers are taking the readouts from the various rings and making sure that everything is maintained properly. This is where some of the gear is stored. You have to put your helmets on because it's you don't want branches falling on people's heads, and the PhD students can have their sandwiches and stuff here. And this is the spot here we have to keep clear where the CO2 is delivered daily to these tanks. And we'll go into here now, and I will flag up a little bit about these tanks. So these are the tanks here, storing, as I say, about 120 tons of carbon dioxide in liquefied form. And I will now click on Nick, and there's a short video, only a minute, that I'll let run, where Nick will explain to you something about these tanks and how they operate. And Nick is the technician who's responsible for making sure the CO2 runs properly and into the rings and so on. The sound may be a little poor on this, so please turn up your volume should you need to.
Actually, Jerry, I don't know about other people, but I can't hear that.
Okay, I'm just gonna stop share. I know what I've done. I need to, when I share my screen, apologies for this, I need to make sure I click the button that shares the sound as well, which I didn't do. Okay. So let me.
The main storage tanks we've got.
Does that work now, Dee?
Yeah,
it's
Sorry, I'll just get it big.
the main storage tanks we've got here hold 120 tons, and it's stored at minus 20 degrees centigrade, 20 bar pressure. This carbon dioxide needs to be reduced in pressure before we can send it to the forest. So the first thing is we're going to take the liquid carbon dioxide, warm it up and expand it, so it then goes from liquid into gas. It comes from our liquid to gas evaporators in through an electric superheater, which is now raising the temperature up to 60 degrees centigrade. Pressure reduced again, and then it goes down to 15 bar, and we're now at 36 degrees centigrade. It then goes into our distribution system, where it's distributed up to each of the array plots.
Okay, if I come out of that, stop Nick sharing. Actually, one thing I forgot to do, and I wanted to show you another video. Actually, it was something I wanted to show you here. Not that one. Not that one. Oh, I can't remember where. Can you remember where it was, Dee? The helicopter shots. Never mind. We'll come back to it if I can remember. Oh, I know where it is. Sorry, apologies. My apologies. It's on the way in. We've not got there yet. So that's what Nick explained about tanks, and these fans for warming up the gas are really important, so that the air and the CO2 go into the rings at the same temperature. Obviously, we've got to keep everything equal. As Nick said, there are 120 tons of CO2 in these tanks. Now, we use a lot of CO2, so approximately 20 tons of CO2 per day. So there's only 5 or 6 days of storage in there, less if it's windy, because you have to pump more CO2 in. And that CO2, if you think about it, it's this 100 kilograms or 1 ton, 1 metric ton of CO2 is what an airline passenger would use return London to New York. So that's basically what we're using every day is 20 individual return flights to New York to get that, to run this experiment. And of course, CO2 is very expensive. CO2 now costs about, the yearly supply of CO2 is over £3 million, because it's an important commercial product that's used in various places. So now we're going to enter into the ring. You can see it's a controlled area, and just like with foot and mouth and other things, we want to keep everything as natural as possible. So both going in and going out, we have to sterilize our boots. And then if we're not lucky enough to get a ride, we have to walk in along the path into the woodland. I'll just see if I can find the video. Yes, so you can see the pylons are huge, and they were insinuated very carefully, like keyhole surgery, by helicopter into the woodland, very, very carefully. So the woodland, which has been there for 180 years, wasn't damaged in the least. And I'll just show you some of this video, and there is no sound to this. So Di and Megan may comment a little bit as we go through. So this is shots of the woodland. And what you can see here is the pylons are not concreted in. They're drilled in on massive screws. And the idea is that when the experiment's over, they can be taken out and there's been minimal impact on the forest at all. And now you're seeing a view from a helicopter. And here's one of the pylons, and it's been winched in by a helicopter and dropped into the forest. So there it's taking off from the field site where they've been stored. This is really...
there's 96 of those in total. I think 16, it takes 16 of those masts to create one of the rings. And then of course you've got the one in the center. And it was pretty much done overnight, wasn't it, Jerry?
It was really quite impressive. Now, this is a bit where I'm really worried about this bloke trapping his fingers, but he seems to get away with it. But this is really quite spectacular. Obviously, you can see it's been done in the winter, which presumably helped with visibility.
I believe the helicopter crew was a specialist team out of Switzerland as well that normally does rescues off of mountaintops.
Look at that, the way that he just puts his spanner in there and then bung. I really want one of those spanners. That's really quite impressive. And there it is. And you can see them standing there proudly. And that's the central pylon as well that was also dropped in the same way. And then just a few shots. I'll just let it roll for a little bit. So you can see there's a central pylon and then the rings around the side, and there's all 6 of them with the central pylon, which we'll finish the tour on in about 15 minutes. Okay, so you really get a sense of the scale of the place, but what a piece of work that was to do that.
I think they are 25 meters high, each of the pylons, and the diameter of each array is roughly 30 meters. And you can see that they're not quite the circle, which you scientists sometimes like everything perfect, but we couldn't quite have the circle we would have liked because they were put in around the trees that were already there. Okay,
so I'll stop that one and we'll continue the tour. It's worth saying that this you could see the oak woodland there in an agricultural area. These oak trees were planted initially as a crop when the Royal Navy would anticipate need for wood for the Navy. So of course that's gone away and so on. So now if we look over here, that was the pipe you saw at the end of Nick's video, and I showed you on Google Earth briefly. That's the pipe bringing in the CO2 to the individual control. So each of the rings, here's the pylons, and this is just the outside of the ring. We'll go in there shortly. This is the control room. So here you can see the CO2 pipe bringing the CO2 into the control room. And if we look inside, what you can see just briefly on there, you can see a map of the actual ring and where all the pylons are. And the CO2 is coming in here, and it's coming in here, and there's valves, and each of these valves then goes to one of the black tubes and is regulated by the computer back in the hut where you saw Nick demonstrating the flow diagram. So each ring has a single control hut that is constantly measuring the CO2 at different levels in the ring and then feeding back from the control center to these valves to ensure that the CO2 level is kept as precisely as possible, 150 parts per million above what the current ambient level is. So if we go inside the face array, remember FACE stands for free air carbon enrichment. You can have a, I'll just briefly have a look around. Obviously, if you miss anything or you feel I've gone too fast, you can always come back and have a stroll through the forest at your leisure. What you can see is on the outside, this is the main ring where, as Dee says, it's not effectively fully circular. But the CO2 is then, or the air, if it's a control ring, is going round, and the blue valves at the base of each of the pylons, we'll probably get a better view of them later, open or close depending on the wind direction to allow the CO2 to blow into the ring. In fact, if you walk into the ring next to one of the pylons, like you can do over here somewhere when you come in, and you put your hand over one of the holes, it just feels like a gentle hairdryer that's blowing in, but it's all the way up vertically. Now you can see, again, because we don't want to damage anything, and I've already alluded to the soil as being something that we think important, exciting things are going to happen in. You can see we've got walkways here, number 6 there for individual ones, but so you can take people in and have a really good look. Initially we didn't have these, but people can come in, school groups and so on, and have a look around without damaging any of the equipment. This is the central pylon that you will have seen from some of the videos, and you can see just briefly it's got ropes and things, and that's where Anna actually climbed the tree every day to do measurements on the top of the canopy. You'll see also there's lots of other plants. There's some ferns, there's sycamores, there's brambles, there's all sorts of things in here, and they will also they're also subject to our experiments, trying to understand what's happening to the CO2 on the ecosystem, not just on the individual oak trees. And that's another reason that why we're doing this at this level. Now, these black pipes here that are strung through the trees, and this white thing here, these are taking in the CO2 and taking it back to the control center and feeding back to the valves to open or close those, depending what they actually detect. I don't know, you can't see me, can you, Dee? Can you see me on screen, Dee? You're muted, Dee.
Sorry, yeah, I can see you. You're small.
Okay, yeah, okay. I was just going to show you if people can see that. That's a CO2 meter that I've got in my office, and it simply measures the amount of heat the gas in the air absorbs, and that's a measure because CO2 is the major greenhouse gas. That's a measure of the CO2. And while we've been sitting here, I've been measuring the CO2 in my office. Of course, my phone has switched itself off because it's I can't even remember my own password. And if we look, of course, it's stopped doing it now. But if I had been lucky, and you would have been able to see. Don't worry, it switched itself off. I forgot my phone was going to switch itself off, but I have got a CO2 measurer on the phone, and that's exactly what it does here. And that's feeding back to regulate those valves that are regulating the CO2 in the forest. So I said about the ecosystem. So here people are looking at germination in quadrats and which seedlings are recruited into the population. For example, some types of plant may do better than others under elevated CO2, and that might change the composition of the ecosystem. But one of the main things is thinking about getting up to the top of the canopy. I've already said that Anna has to get to the top of the canopy, and she did it every day for 2 years in the summer. I did it once, and this is a video of me doing it once, and it was one of the most exciting things I've ever done. So here's a short 1-minute video of me going up a tree. This is the top of the canopy, 35 meters from the ground. Very rarely do researchers get to go this high, but this is where the sunlight is. This is where these leaves will be the most efficient, have the most energy to do that photosynthesis. So this is where the questions have to be asked. So as we look at these trees under elevated CO2, they're now taking in more carbon, more sugar, 30% more than the ones without elevated CO2. And that's what they'll be doing in 50 years' time. We need to understand what happens to that carbon. We need to understand where it goes in order to be able to protect our trees and protect ourselves. So fantastic to be up here. This is a unique experience to be right at the top of the canopy. Hardly ever visited, hardly ever researched, but this is where the action is. This is where the impact of climate change will really happen for these trees. Okay, let me...
You are so brave. You wouldn't catch me up there.
I wouldn't say. I wouldn't say it was brave, Dee. It was just so exciting to go up there, and I think that Anna had to do that, you know, every day. Sometimes starting at 6 in the morning before the sun came up to get that data. I think it's really important to make the point that when you go for a walk in the woods, you see this. Okay, you see ordinary trees and understory and so on. But what's happening is happening at scale is happening at the top of the canopy, where we, as I said in the video, where we very rarely get to go and do research because it's difficult to get to. But those are the leaves with the light and the air and the CO2. The things aren't happening to the same level down here. So we really have to look up into the canopy. And the other thing to think about, we walk in this middle bit, as I say, walking our dogs or just going for a walk in the forest. What's going to happen to that carbon? A lot of that is going to happen under the ground. Trees, plants in general, put quite a lot of carbon into the soil. Maybe 20–30% of the carbon they capture can sometimes go into the soil. It can be through roots growing and dying. It can be through the leaf fall that you see there, but it can also be directly extruded by the roots, and they don't need to do that. So they probably do that for a reason. And we can look at how the roots grow here in this rather innocuous looking black pipe, which is in fact a perspex pipe that's driven diagonally into the ground. And every so often somebody will come along and stick a camera down there. But basically what you then get is a time lapse of how the roots are growing and touching the surface of the tube. And you can then use computer programs to get measurements of how fast the roots are growing. Are they growing more or less, or are they growing differently under elevated CO2? So that's a direct look at that root growth. And there's interesting data starting to come out of that. But we forget also that the soil isn't just dirt. The soil is an ecosystem in itself that we know very, very little about. It's full of microbes, it's full of invertebrates, it's full of fungi, it's full of bacteria that we largely know nothing about. And so what we're also looking at is the soil respiration, because if more carbon goes into the soil and stimulates soil respiration, a soil ecosystem, then the soil might start emitting more carbon in a sort of related way to we're concerned about permafrost melting and putting more carbon into the atmosphere. If the trees overstimulate the rhizosphere, then the soil might start producing more carbon. The balance will certainly change. Now, these little devices, they're not our Star Wars. What they do every so often, they're placed in the rings, and every so often, automatically, this little arm, and you just saw it coming off there, comes over this patch of soil. It's just finishing its job now, measures the respiration, the soil respiration in the ambient and the elevated rings, and then the data is sent back to the center via that ring. So that is also happening automatically all the time as well. I think there's a lot that we're going to find out about the soil that is really, really quite novel. We hear a lot about human health, about the gut microbiome and how it's really important, and people have different gut microbiomes, and the relationship between our gut microbiome and our health is something that people are starting to really understand better, and affecting the way we eat and so on. Looks to me like to us like trees, for them the rhizosphere, the soil around the root, is in fact an inside out gut, and they are looking after that and nurturing that in exactly the same way that we should be doing with our own health. And that's why they're spending some of that carbon potentially on doing that. Okay, so we'll go now to another ring and have a look in that one. And show you some of the other data and experiments that are going on, and just to give you a more complete picture. Here you get quite a good close-up of how these things are screwed in and not concreted into the base there. This is the central pylon in this ring. And again, this is a control ring. You can see this is now contains air, this ring, not CO2. And exactly the same setup, a number of oak trees in here, lots of other different types of tree and shrubs and so on, which are all being looked at by different researchers. A couple of things I think that's really nice to flag up here. A couple of measurements that are also going on continuously. This is a dendrometer, and those of you who know your Greek will know that dendrometer just means tree measurer. And what you can see here is it's effectively a band or a belt around the tree. And just like if you had too big a lunch, you would have to loosen your belt. If the tree grows a bit, the circumference of the tree expands, and we can measure that with this electronic device. And you can see it's got wires coming out of it, and that's done continuously. And so we can compare the growth of the trees in the ambient and the elevated rings, and we get continuous data out of that. Now, if you're interested in the details of that, if you were to go to the BIFoR education site that I pasted into the chat and go to the school's exercise on dendrometer, there's a lot more information and videos on how the dendrometers work, and also some data to download, which schools use in some of their lessons on environmental change and CO2. Dee has just helpfully put that back into the chat. So, of course, the tree is big, which is a fairly obvious statement. But from a tree, or indeed any plant, the problem is that you're doing photosynthesis in your leaves, and so the leaves are doing their job in capturing the carbon. The roots are actually capturing water and nutrients, and you need to communicate between the two. And of course, plants do this through the xylem. And the xylem are basically just tubes which the leaves suck water up those tubes, and they're from the roots carrying minerals, water, and actually hormones and things that actually make up quite a complex signaling thing. But simply, they're just taking water from the roots to the leaves. Now, already talked about stomata. Now, if the stomata are open, you get evaporation from the leaves and water moves up the xylem. If the stomata are closed, such as at night, then there's no evaporation, there's no photosynthesis, because there's no point in trying to do photosynthesis at night when there isn't any light. And so they shut the stomata to save water. And of course, then there will be no xylem transport. So these devices measure how fast that flow of water is going up the xylem, an important component of the water cycle, as I said, combining the carbon and water cycle together. It does this very simply, actually. All it does, it consists of a thermometer and a thermocouple that generates a heat pulse. And so all it does is it heats up for a short period of time, so it thereby heats the water in the xylem. And if that is moving, say it was moving upwards, it moves upwards, and the thermometer detects the time at which the heat pulse arrives at the thermometer, and from the time it took and the distance they are apart, you can calculate the rate of flow of the xylem. And that's an important part of this research as well, because that's part of the story of the tree balancing carbon uptake with water loss. Of course, interestingly, I mean, the xylem, which makes up most of the tree, the xylem is actually the Greek word for wood. So the xylem is actually just wood, and most of it is dead. All xylem is dead, rather, apart from developing xylem, but most of the tree inside isn't transpiring. Most of the transpiration happens in the xylem vessels near the surface, which is why this technique can work. Other experiments going on here. This is a port where we can measure the methane output of the tree under elevated CO2, which obviously methane is another important greenhouse gas. So if the balance between CO2 and methane is changed, that will also impact on the net net amount of greenhouse gases that are around. I also mentioned leaf fall as the carbon rain, if you like, that deciduous trees are producing, and we capture these the leaves in these leaf fall traps. Dee, you were reminding me yesterday of the scale of this and who has to deal with these things.
Yeah, so there's 7 of those traps in each of the 9 arrays. So let me just do some maths. There's 63 of them. And so somebody has to each week during the autumn go and empty each of those 63 traps. And then once they've emptied them, they then have to sort them into all the different species of leaves and whether we've got reproductive outputs, that kind of thing. So it's a huge scale. In fact, a lot of the experiments, if you put one piece of your instrument in one of the face arrays, you have to put it in the others as well. So there's a huge amount of people involved in this project.
Yeah, and one of the things, of course, that's interesting here is if here's a picture of a leaf, you can find an exercise in one of the education links on whether a leaves become more or less infected by fungus, which is a major pest of plants, as you might know if you're a gardener, under elevated CO2 or not. And now this is a shot of a leaf where you can see the trails there. That's an invertebrate leaf miner, which gets in between the epidermis of the leaves and eats it from inside while remaining protected. Now, of course, we have, if the tree has more carbon, then it could potentially, it could defend itself better. It could have more secondary compounds to poison attacking herbivores and pests, or alternatively, if you just put more carbon in the leaf, you might be diluting the nutrients that the herbivores are after, and therefore they might actually eat more. So we have 2 alternatives. Under elevated CO2, you could get more herbivore damage, or you could get less. And that's one of the things that we're starting to look at. Although, Dee, I don't think the data's finished coming through yet, has it?
I think we're very close to publication on that.
So we do have some data coming out, but
yeah.
You'll have to wait for the big reveal. But of course, a herbivore like a caterpillar or a leaf miner is very different from a pest or disease like a fungus, and so they may be completely affected differently. So just because one type of pathogen is affected in one way doesn't mean that others won't be affected differently. And of course, that information, as we said right at the beginning, is really important so that we can understand how to protect forests such as this one under elevated CO2. What can we anticipate will happen, and what control measures or mitigations can we put in place in advance? Just to finish off in this array, here you can see that this is not a managed woodland, which is why I sort of mentioned that you've got to wear a helmet when you go in. If wood falls, we just leave it to rot. And of course, rotting while a tree may capture carbon and store it in its wood, which is a good thing for us in terms of removing carbon from the atmosphere, of course, as it rots and degrades, it will then release that carbon dioxide back into the atmosphere. So these logs, some of them just lying there, and others actually protected from certain types of invertebrates, so we get more insight into the type of decomposition. We're measuring the decomposition of those in ambient and elevated, because changes in the rhizosphere, in the organisms, the bacteria and fungi in the rhizosphere, because of elevated CO2, may affect that process. So that's an important part of that whole carbon cycle. It's not just capturing the carbon. And perhaps one of the most simple devices that we've got, but perhaps one of the most important, is to measure the rainfall, because as I've said, if you take in CO2, you have to open your stomata, and so the drier it is, the the more likely you are to decide to save water rather than to take in CO2. And we call that the photosynthesis transpiration compromise. So you could predict that a tree under wet conditions would take in more CO2 and get more carbon, whereas under droughted years, they might shut down for a bit and save water. So it's really important, even though it's a very simple measurement. So we'll now leave this ring. Obviously, there are 4 others. In fact, there are another 3 that we call ghost rings, which are simply a rope on the ground. And that's another control. We do all the measurements there as well. But there we're trying to just mitigate the control for the effect of all the technology we've dropped into the forest. So effectively, we have 3 types of ring. The one with the pylons elevated CO2, one with the pylons control ambient CO2, and the ghost rings with no technology and ambient CO2, just to make sure we've got most of the bases covered. And this is the central pylon, which is where we'll finish the tour today. And if we go to the top of the tower, you can get a view over Staffordshire, and you can see some of the equipment that is actually measuring the forest. And you can see beautiful day, beautiful spring day in Staffordshire. There's one of the rings looking a little bit like Stonehenge. Some of our English professors, when they were taken to visit BIFoR, made the point it was like looking at War of the Worlds. It is that spectacular. So we'll finish this tour and give us a few minutes for questions and comments at the end with this short video from Rick Thomas, where he's at the top of the tower and explaining to you something about what those devices at the top of the tower do at the level of the whole forest, rather than
got instruments that are measuring things on the woodland scale. So that is, we can see all the trees laid out among us, and we can take measurements that relate to this sort of canopy, to the canopy scale. So the carbon dioxide instrument is over here. It also measures water vapor, H2O, and it does it in a very special way, really, because it measures very, very rapidly, at about 20 times every second. It takes a concentration measurement of carbon dioxide. By coupling that, or particularly the vertical wind component, with the fast carbon dioxide measurements, we can look at how much carbon dioxide is being emitted or taken up by the forest. So I'd like to thank you again for coming along on this tour with us. I hope you've enjoyed it.
Okay, so I will also say I think that's a good place from Rick's comments to stop the tour, and for me to say thank you very much for taking part and listening. And I think we have 10 minutes left if anybody would like any comments or questions or any advice. Remember that that site is open to you just to go back and have another look, to make any comments on, or the school liaison resources. You don't need to be a school kid to go and have a look, but you can have a look at the data and get a really closer look at how some of that kit actually works in the field. And just to say that we're just about to appoint a schools liaison officer, so we're hoping to get that resource and the virtual BIFoR into lots more schools near you. So if you're a governor or a parent or anything, tell your biology teachers about it, and we'll be delighted to try and do visits or talks like this, or actual physical visits to the site. So any questions? If you want to ask a question with your mouth rather than your fingers, then you could put your hand up and we'll keep an eye on that. Otherwise, we'll